Oh, have I done? Okay, all right. Remember that. Don't try that. Okay, why don't we go ahead and get started so that we can get the lecture started on time and everybody get to the rest of their day. Jesse can't be here this morning. He's at an LCME meeting, so he's left me in control of the situation, whatever that means. I like Jesse. I like history a lot too, but I don't know where his source is for finding medical history sorts of things. So all we have to talk about today is just plain history. But there are a couple of cool things. In 1939, on this day, which by the way is kind of a cool day too. It's 10/11/12, which I think is kind of neat. But anyway, so in 1939, Albert Einstein wrote his famous letter to the president, urging the president to back the development of the atom bomb. This came one day after Britain turned down Hitler's offer of peace. And we all know where that ended up. The second thing that I would like to point out was in 1975, October 11th, was the first Saturday Night Live. I think it's kind of a big deal. Now, in terms of announcements, we have one, I think, very nice and very exciting announcement. We had a 100% pass rate on our internal medicine board exam. Thank you, Dr. Casper, and all of the program directors, and thank you to our residents for doing such a great job. It's something I think that we can all be very proud of. So with that then, let's move to today's lecture, and it's my pleasure to introduce our speaker today, Dr. Patrick Gaffney. Dr. Gaffney is now at Oklahoma City, however, did his undergraduate and medical school training in Minnesota, did his residency in Iowa City, and then returned to Minneapolis for a fellowship in hematology, oncology, transplantation, and then a postdoctoral fellowship in, I've forgotten what it was called, but it had something to do with genetics and sequencing and stuff like that, which is what he's going to talk about. He stayed there until 2007 when he was lured away to the Oklahoma Medical Research Foundation, where he currently is a member of the Arthritis and Clinical Immunology Research Program. Looking at his CV, it's very apparent that he has a long-standing interest in trying to determine genetic markers of human disease, and has several interests, including genetic markers of lupus and lupus nephritis. And for those of you who are interested in the topic, he will actually be talking very specifically about genetic markers of lupus nephritis tomorrow at our renal grand rounds, which is held at the KDP building. That'll be at noon in the conference room. This morning, however, his topic is more general. The title of his talk is Clinical Genome Sequencing from Bench to Bedside. It's my pleasure to welcome Dr. Gaffney.
Okay, good. Sound's working. I'm not going to touch that. Thank you, Dr. Lederer, for that beautiful introduction, and I want to thank Dave Powell for helping get this organized and letting me come and speak to you today. It's a pleasure to be here. So today, as the slide shows, I want to talk about a new technology that I think is really going to make an impact on clinical medicine as we move forward into the next decade. And this is clinical genome sequencing. And the outline of the talk today is really, we're going to start with the exploration of the process of exome sequencing to identify a mutation that we found in a genetic disorder from a couple families in our genetics clinic at OU Health Sciences. And then we're going to move to talking just briefly about the historical milestones which sort of led us to being able to do this kind of work, and then review some of the avenues and obstacles that we're beginning to see in incorporating genomic sequencing into clinical medicine, and explore some recent examples from the literature. So I was told that the chief liked to have a case presentation to start medicine grand rounds, and so I'm going to talk briefly. I'm going to present a couple cases for you to get us started today. So the first case is the first family was presented to the OU Pediatrics Genetics Clinic for evaluation of their 24-month-old daughter. Her past medical history included a mild delay in gross motor milestones, but no other real significant information was obtained. On physical exam, the child had a clear spot of cutis aplasia where the arrow is pointing on the top of her head. She also had syndactyly of her second and third toes on both feet, and microcephaly and short palpebral fissures. Her father's history was similar. He had a scar where cutis aplasia had occurred at birth, short distal phalanges in his fingers, absent toes and short metatarsals bilaterally. He also had microcephaly and some borderline mental retardation. And this is the pedigree of that family, showing the, see if I can find my laser, the affected father and then the affected sibling who we had in the clinic that day. The second family was also evaluated in the genetics clinic for a developmental syndrome not otherwise specified. The proband had mild limb reductions of his hands. His sister had shortened distal phalanges of her left hand and bilateral reduction of her toes. And she had a son who also had a cutis aplasia scar from birth, an asymmetric shortening of his hands, asymmetric reductions of his feet, and some intellectual deficits. And this family was really affected. They had 3 generations that had similar features. This is the pedigree from that family, showing the proband here, and then these individuals here that we actually were seeing in the clinic that day. And this is some pictures from that family showing the cutis aplasia scar, some of the defects on the feet. You can see the absent toes here, and it's the shortened phalanges on the fingers as well in the mother and then in the son. So after discussing these 2 families in the case, the diagnosis of Adams-Oliver syndrome was made based on the clinical presentation. And so what is Adams-Oliver syndrome? Adams-Oliver syndrome was first reported as a heritable disorder in 1945. It's characterized by vertex scalp defects, as you've seen on the slides here, and terminal limb defects. The clinical presentation is highly variable, and it's both in terms of its anatomic involvement as well as its severity of expression. The features include, as I've said, most commonly terminal limb malformations, congenital cutis aplasia, which is the second most common feature, also some congenital heart defects, and another finding called cutis marmorata, as shown in this infant here. But this is just a picture from the literature. This was not present in any of our patients. The inheritance of this disease is autosomal dominant most commonly, but it's also seen in a few rare cases as autosomal recessive, and it's been reported worldwide in all ethnicities. There's 2 genes that have been identified as associated with this syndrome, RGAP1 and another gene called DOC6. Both of these genes are functioning in the RAS-GAP pathway and are thought to illuminate a pathway that's important in development of this syndrome. So for the families that we were seeing, we decided to determine if either of these families had one of these gene mutations. And interestingly, neither family had the mutations in RGAP31 or DOC6. So in conjunction with the geneticists at the OU Health Sciences Center, we decided to go ahead and exome sequence these families to try to identify the gene that was affecting them specifically. And I want to make sure that you're clear on this. This is not done as a clinical test. We did not do this as a clinical diagnostic test, but we were doing this as a research endeavor with our clinical genetics group. So what is exome sequencing? Basically, exome sequencing is a process by where we take the 2% of the DNA that's encoding for protein. We isolate it and purify it, and we chop it up into 300 base pair fragments. This DNA is then sequenced in a next generation sequencing instrument on both ends of the strand, about 100 base pairs in on each side. And then these sequences are aligned back to the human genome using a computer to give us a picture of where the sequencing has occurred. Further analyses are then performed to clean the data and then identify variants in the test DNA from the reference sequence. So what does this data look like? Well, this is a picture of what the data looks like, sort of raw right out of the alignment. And what I'm showing here is an exon right here in a gene. And you can see that there's this pileup of short 100 base pair reads that are collected right underneath this exon. And because of the nature of the technique, we do get some spillage or some bycatch that extends beyond the exon. And you can see they're piled up on the forward strand here and on the negative strand here. And in the center, you can see this little green mark. I hope you can see it from the back. It's fairly small, but this would indicate a single nucleotide variant in this exon in a heterozygous state because it's not present in all the reads that are shown here. So this is what we're looking at when we look at exome sequencing data. So what's the process that we use to find the mutated genes once we have our sequenced genes? Well, there are some assumptions that we need to make about the data to process to identify the mutations. First of all, we assume that the mutations that we're looking for in a family like this are very rare. In other words, they're probably not present in mutation databases or polymorphism databases that are currently available. Second, we believe that they follow definable genetic models that can be ascertained by looking at the pedigree. In other words, autosomal dominant, recessive, de novo type patterns of expression. We also make the assumption that the causal mutation is going to damage a protein coding sequence and that it's either going to be non-synonymous, a splice junction, or a frameshift mutation that's going to make some impact, a significant impact on the function of that protein. And that the other assumption is that the causal mutation is likely going to be in a region of high conservation. And the interesting thing about this analysis is that we don't perform any statistical tests. This is purely a filtering of mutations found in our test samples against what's currently available in the public literature. So to do this, we set up using a computer program, a filter and compare type analyses. And what I'm showing is a table here from the paper that we used to describe this. So I'll just briefly go through this. Here's the first family and the second family. And so we looked, first of all, at the number of genes that had greater than one non-synonymous SNP in them. And each individual that we sequenced from those families are shown here. And these are the number of genes we found. We then said, okay, of these genes, how many have at least one of these non-synonymous SNPs that's evolutionarily conserved? And so you can see that the numbers start to go down. And then when you add another layer of filter on here, you say, well, okay, now which of these genes are not found, or which of the non-synonymous SNPs in these genes are not found in the 1,000 Genomes Project, which is a large resequencing project that's looking to find all the variation in the human genome. And the numbers significantly drop when you do that. And then you say, well, okay, of these genes, which genes don't have the non-synonymous SNP that are also in the database SNP 130 from the NCBI, and that goes down even further. And so then we said, okay, of these genes, which ones are shared by the affected individuals within the pedigree? And in family 1, we found 44, and in family 2, we found 26. So then the final filtering step was, of these genes in family 1 and these genes in family 2, which ones were found in both families? And we ended up with one gene, RBPJ. and that's the we believe to be the causal variant or causal gene in this disorder for these families. So what is RBPJ? RBPJ stands for recombination signal binding protein for immunoglobulin kappa region. It's a transcriptional regulator. It inhibits or stimulates transcription based on cellular context, and it's the principal transcription factor for the Notch signaling pathway. And if you remember back to the developmental biology, Notch signaling plays a really very important role in the development of tissues and of cell development and embryology. So we wanted to go back, and now that we think we've found a mutation and the gene that the mutation is in, we want to go back and just look at the raw data just to confirm our beliefs here. And so I'm showing you one of those plots again, except we're zoomed in on the reads. And you can see here, here's the affected father in that family 1, where you have this mutation is shown here in RPBJ, and about half of his reads in this region show an alternate allele compared to the wild type. Here's the mom who has a homozygote for the wild type allele, and here's the affected offspring who is again heterozygote for the mutation in this exon in RPBJ. So that data looks good and clean, and so I think we made the conclusion that this was probably likely the causal variant in both of these families. So where are these mutations located within RPBJ in these families? And so you can see that when you line up the amino acid sequence of RPBJ and you compare it across multiple different species, you can see that, and I've outlined in this black box where the polymorphic mutation occurs, and you can see that it's in a region that's highly conserved across species, and this is in the first family, and here's in the second family. It's in 2 different locations within RPBJ, so it's not the same mutation or the same amino acid residue that's mutated. It's 2 different residues, and both of these regions of RPBJ are shown to be in a region that, when you line up the 3-dimensional molecule, in a region that's thought to interact with the binding groove of DNA. So I told you that RPBJ is a transcriptional factor, transcription regulator. So it's really important for it to interact with its binding sites on DNA to initiate gene transcription. And it was interesting to us that both of the mutations in each family were in a different region of a part of the protein that interacted with DNA. So we assumed that, we hypothesized that these mutations may interfere with RPBJ's ability to bind DNA at certain times in developmental biology when it's important for development of the limbs. And so when we did this, we did an in vitro assay, just a gel shift assay, and we were able to demonstrate that compared to the wild type, both protein, the RPBJ expressing the one mutation from one family or the other mutation from the other family had impaired binding of to DNA compared to the wild type. So that was the functional evidence that we needed to support this observation. So in summary, for these 2 cases, we had 2 independent families with clinical presentation of Adams-Oliver syndrome, with really no evidence of the known Adams-Oliver syndrome mutations. We decided to perform exome sequencing to identify new mutations in these families, and we found 2 unique mutations in the Notch transcription factor, RBPJ, in each family. And this RBPJ mutation is likely the causal mutations in these families because it interferes with the ability of RBPJ to interact with DNA. And what's really interesting about this is that from the time that we got the DNA in the lab to the time that we had a gene in hand was roughly 2 weeks. And for anybody that's lived through the linkage years, when we were studying single gene disorders using linkage, it would take years and lots of genetic mapping to come up with this kind of a diagnosis. And so for us, having worked with complex genetic diseases like lupus, this is a real hoot because we can make a gene discovery in a matter of a few weeks using this technology. So I think, and this has really become a common scenario in Mendelian disorders using exome sequencing. And so much so that when you look at the literature over the past few years, you can see that the papers that are coming out with exome sequencing in Mendelian diseases is skyrocketing from year to year. And even then, just in general, exome sequencing is increasing on a year-to-year basis. But now we're starting to see even more papers coming out that have a clinical piece to them. In other words, the rapidity with which you can get these diagnoses is starting to become relevant for clinical, real-time clinical applications. And so we're starting to see papers come out that are using actually these technologies for real clinical diagnosis. And that's sort of where we're going to go with the next part of this talk. But I think it's important before we go there to sort of figure out how did we get here? How did we get to this point where we can really do this? And Dr. Lederer came up and gave us a little bit of a history lesson before the talk. And so we're going to go back a little bit and explore some of the genetic foundational milestones that in genetics and genomics has really allowed this technology to get to us today. First, I'm going to start with Gregor Mendel, who, with all of his experiments, really sort of defined the laws of inheritance. This was followed a few years later with Friedrich Miescher, who really defined or discovered nucleic acid. And then in 1944, Oswald Avery and his group discovered that the DNA was actually the molecule or the material in the cell that transmitted heredity in a very famous experiment called the Oswald experiment. And then that followed up in 1953 with the discovery of the structure of the double helix by Watson and Crick. And then after that, the next big discovery came in 1961 with Marshall Nirenberg at the NIH, who really then decoded the DNA code, understood that the triplet-based codon was what dictated what protein residue was going to be added during a translation of mRNA. And then being able to read the DNA code, Frederick Sanger and his work to develop the Sanger sequencing method really really set the stage then for what has followed beyond that, which is in 1990 the Human Genome Project. And so the Human Genome Project took off in 1990, and the goal of the Human Genome Project was to sequence the entire human genome. And this set up perhaps one of the greatest, I guess one of the greatest scientific rivalries that we've seen in at least my memory, where you had Francis Collins, who was leading the effort for the public sequencing effort, and Craig Venter, who was leading the private sequencing effort. And they were using instruments that employed Sanger's method for sequencing the human genome, and in laboratories all around the world that looked much like this, huge operations where multiple sequencers were working 24/7, generating data that looked a lot like this. It took them roughly 10 years, and the first draft of the human genome was published in 2001. And then beyond that, it took a couple years to go back and really go over some of the difficult regions of the genome. Not all the regions of the genome would give up their sequence as easily as others. And so they went back and kept going back to try to fill in gaps and sequence regions that were very difficult. And in April of 2003, the final sequencing project was declared over, and the Human Genome Project was deemed finished by those individuals that were leading it. And so this began lots of conjecture and speculation of what we're going to do with this human genome sequence. And in the popular press, there was lots of discussion about the dawn of genomic medicine and what is genomic medicine. And this wasn't isolated to just the popular press. It was also picked up in the scientific press in discussions about what genomic medicine really meant, and the glowing DNA and the beaker and all of this. And so I think it's important to really sort of define what is genomic medicine. And the best definition that I've been able to come up with is one that Eric Green has described. And he has said that genomic medicine really is healthcare tailored to the individual based on genomic information. And so if that's our definition, then how do we get there? How do we get to the realization of genomic medicine? The Human Genome Project has really laid the groundwork. This was really the race, I guess, what you might say, the race to the starting line. And now we need to figure out what it's going to take to get us to the realization of genomic medicine. And there's going to be lots of ways to do this, and exactly how we get there is not entirely clear, but I'm going to go over some of the major issues and major efforts that are underway to get us to that point. And so the first one, I think, is trying to understand what the function of the genome is. We have a pretty good idea of the function of the 2%, the coding region, that codes the proteins that are important for doing all of the things that proteins do. But there's lots. What about the other 98%? And so there's lots of interest in this and lots of efforts going on. For one is the epigenomic landscape. There's lots of research going on in this area. The epigenomic landscape has a lot to do with what's going on, not so much for the coding, for things that change the coding of the DNA, but for modification of the chromatin, a modification of the genome through processes like histone modifications with acetylations or methylations of DNA that alter the transcriptome and alter the chromatin in such a way that genes are expressed differently. The other major project is the ENCODE project, and I don't know if any of you have noticed in the last 3 weeks the ENCODE project dumped 30 papers into the literature at one time, and they set up a website. So they anticipated that everybody would have a little data overload, and so they set up a little website here where you can actually go and navigate through all of that literature that they have dumped into the literature at one time. And it's really an amazing amount of work. But what the ENCODE project is designed to do, and what they're focusing on, is developing an encyclopedia of DNA elements. And that's what ENCODE stands for. And they have discovered that 80% of the non-coding DNA really does something. And what that something is it's sort of a molecular circuit board for orchestrating different processes in the cell for transcription of DNA and transcription of proteins. And what you see here is that the ENCODE project has discovered that these non-coding regions are very critical for defining patterns of gene expression, for determining context of cells, for development of different cellular structures, and for dynamics of the DNA, the chromatin dynamics that are going on at various stages of the cell in response to outside stimuli, as well as other cellular functions. And so this is really a critical piece of data that is being now very much enriched with the latest set of papers that have come out. The other thing is we need to understand more about the human genome variation, the 0.1% of our DNA that actually is different across populations. 99.9% of our genomes are identical by sequence, but it's really this 0.1% or about 300 million bases that are variable that can tell us volumes about human disease. And so there's been large efforts to build an encyclopedia of human genome variation. And one of these has been the HapMap project, which is designed, and the goal of the HapMap project is to identify all of the haplotype blocks, various haplotypes that are present in our DNA that span the entire genome across multiple populations. The 1,000 Genomes project, which I mentioned earlier, which is going through and sequencing thousands of individuals from different populations to try to catalog all of the variation that we see in the human genome. And then the other project, the UK 10K project that's going on in Europe. So then, understanding the genomic basis of human disease, using some of these databases, this information that's being generated to understand the genomic basis of human disease is another critical important piece to this. And the genome-wide association study, that technology has really been the leader in this area. And I think that this has really been a breakthrough for us in complex disease genetics to be able to take groups of patients and well-matched controls, take their DNA, put them on microarrays that have single nucleotide polymorphisms on there, and then identify with extreme statistical significance associations of genes that are enriched in the cases versus the controls. And this has really been a breakthrough for complex diseases. And the first GWAS success story was really published several years ago and showed that complement factor H polymorphism is a cause for age-related macular degeneration. And this was the first publication that really showed this technique could work. And since then, there's been a massive explosion of GWAS, as everybody who has a phenotype is wanting to do a GWAS in the phenotype. And you can see in this figure, it's something kind of hard to even look at. These are the chromosomes. This is from the NHGRI website. These are the chromosomes, and each red or each circle represents a significant genome-wide association gene identified for, and the color of the circle is the specific disease that has been identified. So there's multiple hits across the genome, multiple diseases that have been elucidated using this technique. And so at this point now, I think where we're going forward here is to start to get routine genome sequencing into clinical practice. And I think that's what's pushing the field forward. But how do we do that exactly? Well, at the completion of the Genome project in April of 2003, the NHGRI, Francis Collins and his group there, wrote an article in Nature, and they said that technological leaps that seem so far off as to be almost fictional, but which, if they could be achieved, would revolutionize biomedical research and clinical practice. For example, the ability to sequence DNA at costs that are lower by 4 to 5 orders of magnitude than the current cost, allowing a human genome to be sequenced for $1,000 or less. So this was the first indication that in order to get to clinical genome sequencing on a routine basis, the cost is going to have to come down. And so there's been a tremendous amount, since 2003, a tremendous amount of effort to develop the technologies that are going to get us to this more affordable genome sequence. And these are the instruments that are now out there that have really started to change the landscape for clinical genomic sequencing. And all of these are clinical sequencing, are instruments that we use in the lab from various vendors. And the most recent one, I should point out here, not in the corner, is a sequencer that looks not much bigger than a jump drive that you can actually plug right into your computer. If you were doing field sequencing out in, let's say you're doing crop genomics or something and you wanted to sequence something out in the field, it has a little port here where you can actually insert your DNA, and it will sequence the DNA right on your computer. So that's where we're headed. And so all of these new technological breakthroughs have really started to drive the cost of sequencing down. This is another slide from the NHGRI, and you can see the classic Moore's law curve, which originally said that the number of transistors on a microchip will double about every 2 years. And so this is the curve that defines that. You can see that the cost at the time that these sequencers started hitting the market, the cost of the genome to sequence a genome has plummeted. And about this time today, our cost for sequencing a human genome is roughly around $7,000 or $8,000, depending upon what vendor you use or what your current laboratory costs are. I should note that it cost the Human Genome Project roughly $1 billion to sequence the first human genome. So we've made tremendous progress in about 10 years. So the ability to do this, however, has only elucidated another bottleneck, and that is, how do you interpret and manage all of the data that these instruments put out? And so this becomes a real problem right now in the path of genomic medicine, is an analysis of this data. And again, the literature has jumped on this. There's lots of discussions about how we're going to handle all of the data that's coming out of these sequencers and how that data is going to be analyzed and brought into the clinic so that it can be useful and meaningful. And on the celebration of the 10-year anniversary of this Human Genome Project, Harold Varmus wrote an article in the New England Journal of Medicine, and he was quoted in this article saying that physicians are still a long way from submitting their patients full genomes for sequencing, not because the price is high, but because the data are difficult to interpret. And this is sort of a cartoon I found not too long ago, which sort of illustrates that. Where the two docs are at the bedside of the patient, and they're trying to read the genome, and he's scratching his head, and she's got her arms thrown up. They don't know what's going on. And so what's the source of this bottleneck? Well, I think it's multiple areas. First, it's probably in hardware. These data sets are huge. Just moving the data from one site to another, moving it from your sequencer to your sequence storage facility, moving this out of the storage and into a computer that can actually analyze it is not trivial. And so there's lots of work going on trying to figure out what are the best ways to manage that. Software is another issue. We have to have better software to analyze the data, faster software, more efficient software. And then in personnel, these sorts of data need highly skilled and trained bioinformatics people. But the bioinformatics people need to understand the biology to some extent, and there's a real need for doing that. However, I wouldn't say that all is lost. I think we're all making progress in all of these areas, and I've seen just in the few years that we've been doing this sort of work, a substantial improvement in many of these aspects. So what I want to do now is I want to show you that this kind of stuff is already happening. It's appearing in the literature. People are using these genome sequencers for specific clinical questions. And so we're going to go through a few clinical, examples from the current literature where this has been applied. And I think one of the most interesting ones was this paper that came out in 2011 by Elizabeth Worthy and her group, where they used exome sequencing in a child that had intractable inflammatory bowel disease. This, if you read the paper, you can just sort of understand the frustration and the difficulty that these physicians had in trying to get a handle on what was going on with this child's disease. They had a 15-month-old male who had inflammatory bowel disease, and the child was refractory to all standard therapy and was continuing to fail to thrive. A very, very complete and extensive workup failed to identify a causal mechanism of this child's disease. And so they ultimately said, we don't know what it is. It looks like it's genetic. Let's do an exome sequence and see if we can discover something that might help us understand the biology. When they did that, they found a hemizygous mutation in a gene called X-linked inhibitor of apoptosis. And this gene was de novo in this child, and they referred this child, based on this finding, for a hematopoietic stem cell transplant. And you can read the paper, but there's a lot, this was not an easy decision to make, obviously. But they went through and, based on this finding, they decided that this hematopoietic stem cell transplant was likely going to be the only way that they were going to be able to correct this deficiency. And when they did that, once they got the kid through the stem cell procedure, the symptoms significantly resolved, and the child is doing okay. So this is really the first example of where this was actually done in a clinical setting. Now, it wasn't done, in a rapid turnaround fashion, and it was done through a very, very specific advisory board type mechanism. But it was a very interesting example, and if you ever hear these guys tell their story about this, it's quite an interesting story. Drug repositioning is another area that's getting really heavily looked at with this. So here's an example from a recent paper a few weeks ago where they were doing a trial of everolimus for bladder cancer. And the study didn't meet its disease-free survival endpoints, and so it was determined that everolimus was not that effective for bladder cancer. However, what they did notice was that one patient in the study had a complete durable remission, shown here where he's got lymphadenopathy and pre-treatment, and then post-treatment, all of that lymphadenopathy has resolved. And so they were interested in figuring out, well, what's unique about this person? So they did an exome sequence on this person, and they did a whole genome sequence on this person, and they found a frame shift mutation in this person's tumor in a gene called TSC1. Interestingly enough, TSC1 was in the pathway targeted by everolimus and made the tumor exquisitely sensitive to everolimus. And what they also noticed was that there were 3 other patients that also had not complete remissions, but partial remissions of their disease. And when they went to sequence those individuals, they found different TSC1 mutations in those patients. So it suggested to them that if we knew that information beforehand and we did a clinical trial, they would have much better success at demonstrating an effect for everolimus. So here's another example where we're understanding these mutations, and using this technology can really help us treat the right patient with the right drug. And so here's another one, infectious disease. Now in this case, it wasn't the patient that was sequenced, but it was the organism that was infecting the patients that was sequenced. And this was a study in the New England Journal where they used rapid whole genome sequencing for investigation of a neonatal MRSA outbreak. And so sequencing was used, as I said, in this NICU outbreak of MRSA. And what they were able to determine using whole genome sequencing of the organism was that they could distinguish between the outbreak and non-outbreak isolates, and that the whole genome sequencing was better than an antibiogram in determining which of the isolates were from the outbreak strain versus the non-outbreak strain. The sequencing also confirmed the observations of the infectious control team in identifying the transmission events at the outbreak, and they speculated that if they had this information prior to the outbreak or more during the outbreak, they could more accurately make decisions regarding the need for their ward closure. They ended up having to close this NICU for about a week to decontaminate everything and to try to control the outbreak, and that was a significant issue because of just the issues with having to move patients and to send patients to other hospitals. And so this is another example where in the setting of infectious disease where this technology can be quite useful. And here's another interesting story. This is the story of personal omics that was reported by Michael Snyder, who's at Stanford University in his group. And basically what he did is this was the scientist as self-experimenting on himself. And what he did is he subjected himself to a 14-month exploration of his personal omics profile by serial blood draw and various other testing that they did on Dr. Schneider during that time. And so what he had done was genomic, transcriptomic, proteomic, metabolomic, and autoantibody profiles over a 14-month period on himself. And what was interesting was that he found through his genomic study, through his genomic sequencing, that he was at a significantly elevated risk for type 2 diabetes. And his lifetime risk, based on his genetic studies, and I know you can't read this, I apologize, but he was at least a 50% lifetime risk of developing type 2 diabetes. He didn't have it at the time that he started looking at this, but the risk was high. And so he convinced one of his colleagues to say, well, my genetics tell me I'm supposed to get type 2 diabetes. Can you just check my blood sugar during this time frame while I'm doing this? And the colleague was all, that's not going to help anything. But he convinced her to do this, and so she did it. And believe it or not, he got an RSV infection about day, I want to say 300, or about day 300 into this study. And right after that RSV infection, he started to see an elevation of his blood sugars. And they noticed it for several days, and finally, eventually, confirmed that he really did develop, while he was observing himself, type 2 diabetes. And he underwent a series of treatments for this, and the blood sugars and the hemoglobin A1C levels began to come down over that time. But he was actually able to watch himself develop type 2 diabetes in real time, which I don't think I've ever seen before, which is quite interesting. And of course, the blood sugar responded to his lifestyle changes and whatnot. So here again, another indicator that some of these technologies can be quite informative and maybe even predictive. The last example would be in cancer mutation profiling. And I think the cancer field is way ahead of most every other field in this area, as exampled by the drug repositioning study I showed you. But there's huge efforts now to comprehensively characterize all sorts of different common cancers that are afflicting our population. These two papers just came out, one in human colon rectal cancer and the other one in squamous cell lung cancers, that characterize and comprehensively identify the mutational spectrum. And I think from these databases, we're going to really be able to understand better subsets of cancers, their mutational profiles, and their behaviors clinically. And this, I think, is going to eventually be useful in both drug design and patient management. So I think the path to genomic medicine is progressing. It's knocking at the door It's not there yet, but we're getting very close. Now the things, in the last couple minutes here, I want to just bring up another issue that we really haven't talked about, and I don't have a lot of time to talk about it, and that's the issue of the ethical and the legal and the psychosocial implications. These are issues that are going to need to be worked out before this really becomes common practice. Issues of informed consent. How do you inform consent properly for these types of tests? Privacy of the data. How do you adequately store the data and privatize the data so that the data can't be used against HIPAA violations and things like that? How do you determine what results you're going to return to the patient, and especially with respect to incidental findings? So, for instance, if you do an exome sequencing scan in a person for a given indication, and you find that that person has a mutation for a high likelihood of developing BRCA1 breast cancer in their lifetime, but that's not why you did the test, what's your obligation to return that information to the patient? And how do you prepare the patient for that information should they want it? These are all issues that need to be discussed and worked out clearly before we have this kind of testing going on routinely. Access to insurance. Access to health insurance is protected by law, but what was interesting was that in the Michael Snyder case was that when he discovered that he had a high genetic risk for type 2 diabetes, he wanted to get life insurance, and the life insurance quotes that he was getting were astronomical based on that information. And so those are issues that we're going to have to think about, and things are going to have to be dealt with in that area. And also the impact on family members. So if you do genetic sequencing on an individual in that family, the offspring, the first degree relatives, are going to be related. They may have a chance of having the same problem, but they may not want to know, or they may have a different thought about what they want to know about that information. And so those are issues that are going to have to be talked about. And you're fortunate here because you have an Institute for Bioethics, Health Policy and Law that's been very active in this area. And so I think that when I just wanted to call out this group, because I think there's been really good information that's been put out there that's been driving the discussion from your group here at Louisville. So I think the future is bright for this area. Genomic medicine is going to be a very exciting area. It's going to impact dramatically the way we all practice medicine going forward into the future. And I just hope that I've been able to portray to you today some of the exciting potential of this technology, but yet give you some sense for where we've been and where we need to go yet. So in summary, we've gone through the milestones in genetics. We've talked about the Human Genome Project, the databases that have been come out as a result of the Human Genome Project that have been very critical for our understanding of genomic biology, the advances in genomic technology, the rapid high-throughput sequencers that are now allowing us to do this kind of work, brought up the issues of dealing with the data and how do we get to getting to really clinically relevant results that can be used in the clinic. And some of the early examples of potential clinical impact of this technology. And then just briefly touched on the ethical, social, and legal implications of the work. So the way forward, I think, is yet to be determined, but I think everybody in this room hopefully will have some impact on that area. And I want to thank you for your attention today and just go through the acknowledgments for my group. The Adams-Oliver work was performed by Graham Wiley, Stuart Glenn, Xiaofeng Wang, and then my collaborators at OUHSC, Susan Hassett, John Mulvihill, and Klaus Weiringa. The work was funded by a OMRF grant, and then I want to thank Eric Green for his inspiration for some of the slides that I used today, and we'll go from there. Answer any questions you have.
Well, thank you for such a very enlightening discussion of something that I think has been kind of murky for many of us in the literature. I'll start off with one question. For the families that you looked at that had this unusual syndrome, what was their reaction when you found out what the gene was?
Yeah. Well, one of the families was lost to follow-up, so we haven't really been able to make contact with them. The other family was pleased to know that we had a gene that was identified in their family. I think that they were just curious to understand what was causing this in their family. You know, I don't. At this point in time, there's probably not much more that we can do with that, except let them know, if they were, if they were a carrier, that there's a chance that they're going to pass this on to offspring. But I think that that's our limited experience in that area.
Okay. Any questions? Yes.
Thank you for your talk. It's wonderful. I just had a question about what we do with this information, and I know in that one instance of the child in the short bowel disease that the answer was transplant. And of course, there are a lot of risks and lifetime complications associated with transplant. Can you speak about, or just give a brief overview of anything else that's being done in this world, and maybe turning off certain the expression of certain genes with certain drugs and the development in that area?
Yeah, I think the way some programs are addressing, who to sequence and what to sequence is, and this is what I've seen most commonly at places that are actually doing this, is to have a scientific advisory board, essentially, that will review cases, and based on input of bioethicists, geneticists, clinicians, multidisciplinary teams, they make a decision about this particular family has a high likelihood that exome sequencing or whole genome sequencing will be helpful in the diagnosis, or this particular patient, given the clinical situation, will have less likelihood. And so there's sort of a multidisciplinary approach, I think, is what most groups are using to try to get patients, to try to figure out who's the right group to apply the technology to at this point in time.
Jeff?
A related question. Given the explosive growth in genomics that you described, to my knowledge, the interventional whole cases are relatively small. And I'm just wondering, is there projections on the visible horizon, beyond putting a label on these things, how many will actually be something we can impact on?
Yeah, I think right now the real, and this was the case for when we were studying, genetic diseases way back in the 1980s, and we made a lot of progress in Mendelian disorders, the rare disorders, using linkage analyses. And then when we tried to do the same in complex diseases, which is what most of what we see in the clinic, like heart disease and diabetes and rheumatic disease and those kind of things, it's much more difficult. And I think what you're seeing is sort of a part two of that here, where you see the technology really having an impact in the single gene disorders, but the impact in the complex disorders that have probably a polygenic nature to them is going to be slower. But I think I think a lot of this is going to also be in trying to figure out subsets of patients with complex diseases that may have specific genetic drivers of those diseases that when you look at the whole disease as a whole, you wouldn't necessarily be able to pick that out. But if you look at a subset of patients with unique phenotypes or extreme phenotypes, you may be able to start teasing it apart that way. It's going to still take a lot more research, but one of the things that we've been thinking about doing is basically seeing if we could develop a tool, an exome data tool that would impact things in autoimmunity. First of all, the three things that we've targeted would be sensitivity based on pharmacogenetic data. So we can sequence and find pharmacogenetic polymorphisms that we know modulate rheumatic disease drug metabolism activity. That would be one thing that a clinician could potentially act on. We're developing a kidney nephritis risk panel, and so we're trying to figure out a way that we could screen a potential patient who doesn't have nephritis based on their genetic profile of what their risk for developing nephritis would be. That could alert a physician to, making sure that they keep them off drugs that may impact kidney function, follow their renal activity more closer, that kind of thing. And so the other thing is looking at, for rheumatic diseases, patients that have high risk for comorbid illnesses, cardiovascular disease, things like that might be at a higher prevalence in a patient with lupus, let's say, that we would want to make sure that we get out ahead of, and we're trying to see if this information might be useful for doing that kind of work. But it's still very early on.
Okay. Thank you very much.
Okay, thank you.